Okay, so three wonderful speakers. This will be, I think, very dazzling for all of us. I'll start with Ravi Goyal. He's an assistant professor in the Division of Infectious Diseases and Global Public Health at UCSD. His research interests lie at the intersection of statistics, network science, and implementation science. His research contributions have been the development of novel statistical network analysis methods that aid in designing and assessing the impact of HIV and COVID-19 interventions. And next, we have the esteemed Dr. Natasha Martin, an infectious disease economic modeler and a professor in the Division of ID and Global Public Health, also at UCSD. She co-directs the NIDA-funded T32 program in substance use and HIV and the UC GloCal Health Fellowship. She is also the co-director of the Biostatistics and Modeling Core of the San Diego CFAR. Her primary research focuses on modeling hepatitis C and HIV transmission and prevention among key populations, such as people who inject drugs. Her modeling work has informed several WHO guidelines, including when to start ART in people living with HIV, and the WHO Global Health Sector Strategy on Viral Hepatitis. And finally, Niema Moshiri is an associate teaching professor in the Computer Science and Engineering Department at the University of California, San Diego, UCSD. He works on computational biology with a research focus on viral phylogenetics and epidemiology. He also places a heavy emphasis on teaching, namely on the development of online educational content, primarily massive adaptive interactive texts, which I hope we'll learn more about during this presentation. I'll pass it on to you and assume that you will be sort of eyeing the chat, but I'll keep an eye on it as well.
Yeah.
Welcome.
Thank you so much, Jill, and thanks everyone for coming. It's nice to see so many people here on Friday morning. We are really excited to present our work. Before I get into that, sorry, but I wanted to do a little bit of advertisement for the San Diego CFAR Biostatistics and Modeling Core, of which Ravi Goyal and I are in the leadership for that. Just in case people haven't heard of us, we are a resource for consultation, collaboration, and training related to quantitative methods applied to HIV, in particular biostatistics and sort of infectious disease modeling. We have an expertise in a wide array of studies, lots of different specialized analyses that we can provide input on. We provide pre-award grant proposal support if you're developing a plan and you need some kind of guidance on the analytics. And we also provide collaboration on funded proposals, all of this, of course, related to HIV. So if you need assistance, we would love to work with you. Please go to the website and submit a service request, and we'd be happy to chat. All right, so moving on to what we're talking about today. I know it's unusual to have three presenters, but I think it accurately portrays kind of the collaborative group effort and heavy quantitative lift that this project has entailed. We really could not have done it without the team that we have, and each person provides a really unique area of expertise that has sort of melded together to mean that this project in particular, of course, we're always motivated by the public health impact of the work that we do, but has been unique in its innovation in quantitative methods. We've really had to kind of push the boundary of available and new methods in order to get this work off the ground. And so I think that culmination is what has made this project really near and dear to my heart. So this is work that was funded and started under the Triumph project, which is led by Susan Little and her team. And the aim of our part of it was to improve HIV incidence estimation through the integration of molecular data with epidemic modeling. And this work that I'll be talking about today has been applied to San Diego County. So just some acknowledgements, as I mentioned, this is a large team. We are using San Diego County data, so a lot of people on the data analytics side helping to process that data and provide us inputs, as well as people helping to think about the quantitative side. And we're eventually applying this work to a separate NIH-funded grant with the Florida Department of Health. So we have a large team working on that as well. All right. So, of course, everybody here knows about the Ending the HIV Epidemic goals. And in particular, one of these goals is to reduce new HIV infections by 75% by 2025 and at least 90% by 2030. What this means is that our main output that we're interested in is new HIV infections, but it requires kind of estimating how many new HIV infections we have today and predicting how many new HIV infections we'll have in the future to understand whether we're on track to reach those goals. And it also requires us to understand how can we optimally allocate the prevention resources that we have today to reach those incidence targets, to both sort of maximize the impact of the resources that we have and also ensure that we have equity achieved between our populations. And I won't talk too much about equity today, but it's a very strong component of the work that we are doing and we plan to do in Miami-Dade. Thinking about both how we reach these targets, but also how we do that equitably. All right, so where does epidemic modeling fit within that? And what is epidemic modeling, I suppose? So epidemic models are a type of simulation model that mechanistically simulate infectious disease transmission patterns. Now, the mechanistic word is really important here because what it means is that we're trying to develop simulation models that incorporate what we know about how an infection is transmitted to mechanistically reproduce the history of the epidemic as observed and to use that to predict into the future. And so these models are useful in estimating quantities that are hard to measure. For example, true incidence, when we know that there are, for example, people that are infected today that are not diagnosed immediately. And so, we don't have a good estimate of true incidence, but they can also be used to predict into the future. And in particular, they're used often to as a way, as sort of a test case way to simulate the potential impact of different scenarios or public health interventions or policies to say, well, what could we expect if we did X versus Y? And overlaid onto all of that is that they can be helpful in informing resource allocation. So not just what happens if we do X versus Y, but how much would it cost to do X versus Y, and what are the relative costs and health benefits if we do a particular strategy, and what is the cost effectiveness of that strategy? Is the additional cost worth the additional spend? So as I discussed before, one of the main outcomes that we're interested in is HIV incidence, both today and in the future. And it's very difficult to measure current HIV incidence because of diagnosis delays, and some of the methods that have been developed to estimate that assume sort of statistical methods or other types of modeling methods may have to make assumptions that testing patterns are staying the same over time. And we know that that's not necessarily true overall, and particularly among subpopulations. And so it's been difficult to develop kind of robust ways to measure current HIV incidence. But additionally, when we develop sort of prediction models of future HIV transmission and incidence, such as using the mechanistic transmission models that I've been talking about and that our team uses, that kind of prediction modeling is often hampered by a lack of data, and in particular, a lack of robust data on understanding how our populations are mixing. And when I mean mixing, I mean sort of sexual mixing for sexual risk and choosing sexual partners, as well as, for example, injection partners, and how that mixing varies by risk group, by race, ethnicity, or by geography. So these, as you can imagine, sort of differential mixing patterns by these characteristics will affect how an infection sort of spreads and the impact of interventions which are targeted at particular groups. And therefore, if we make incorrect assumptions about mixing patterns, it could possibly lead to both incorrect intervention targeting or suboptimal resource allocation. So these are the existing challenges that we have with the methods that have been currently developed, among other challenges, I will say, but two key ones. All right, so the sort of the sort of thrust of this project that I'll be talking about today is the concept that I'm introducing on this slide. And that is that molecular data likely contains additional valuable information that we could be using to inform our mechanistic epidemic models, which, as I mentioned, are therefore used to predict the epidemic as well as do downstream resource allocation. And so traditionally, these epidemic models are usually informed by epidemiological data, for example, just routine surveillance data and new diagnoses, maybe cross-sectional or longitudinal studies looking at HIV prevalence overall and among particular key populations, et cetera. And they can also be informed by behavioral data. So, for example, self-reported data on sexual and injecting risk from cross-sectional or longitudinal surveys. That's sort of the traditional type of information that's used to parameterize, to kind of feed into these models, but also calibrate. So ensure that the model accurately fits the observed data that we see. Our premise is that molecular data, so for example, phylogenetic trees and cluster characteristics, potentially provides really valuable information on transmission patterns that could be used to additionally inform our transmission models, but have mostly not been incorporated into these epidemic models historically. So on the right-hand side here, you can see an example of the sort of molecular network and connections. So these are viral genetic connections between individuals, the different colors and shapes representing different risk groups, and I think race, ethnicity. But anyway, you can see that there is information in this network that is probably very valuable in terms of our understanding of mixing patterns and groupings between transmission between individuals. So to date, there have been a few very limited examples of these epidemic simulation models incorporating features of the molecular network. And this has been done relatively recently and just by a handful of individuals. The reason why this hasn't been more widely adopted, there are a few challenges. One, that we kind of require ongoing methods development in terms of figuring out how to properly incorporate these data and how to incorporate them into our kind of end-to-end simulation of the epidemic and then down to the molecular networks. And that has been hampered because they're extremely computationally intensive and are a huge computational burden in trying to run these models. And finally, the observed molecular network that we have is incomplete, and we have non-random sequence sampling in terms of who is sequenced. And so it's very hard, traditionally it's been very hard to draw inference on that, broader molecular network when we have this incomplete and non-random sampling process. So these are kind of through a few challenges that we have had to tackle on this work. So our key question that we're trying to answer here is, can the integration of molecular data actually improve our simulation model estimates of HIV incidence in order to inform prevention efforts and resource allocation? We truly don't know the answer to this question. Well, we didn't when we started. We thought that the integration of molecular data would be an improvement, but again, this was really, something that we had to test in our process. And so we had three key methodological challenges, and this talk is where we kind of split the talk between the three of us, because each one of us has had to sort of tackle one of these challenges. And so we'll start with Niema, who will talk about how we have tackled the first challenge, which was really a method challenge of how we do this. So how do we create a simulation engine that simulates the contact network of individuals, HIV transmission on that network, and then the phylogenetic network on top of that? And we needed to be able to do that in order to compare that to the observed epidemiological and molecular data to ensure that we had a good match. And then the second key challenge was this issue that I mentioned about the incomplete viral genetic network that we have. And so we have this partially observed viral genetic network, and we have non-random sequence sampling among our groups. And so how do we handle that issue of missing data and and missingness that's not equal between our groups. And Ravi will speak to that methods work there, where he's done a lot of really innovative work. And then one of our third challenges, which I would say is still ongoing, is trying to understand, well, what are the best calibration metrics that we'll choose from the molecular data in order to inform our epidemic simulations? Because as I mentioned, there's just not been a lot of work in this area, and there's a lot of different metrics that we could choose. So we've had to think about that a little bit. All right, so now I'd like to turn it over to Niema. Niema will advance the slides. We'll talk about how we've tackled this first challenge of this sort of simulation of this whole process. Niema?
Awesome. Yeah. So as Natasha mentioned, kind of the first step is just how do we simulate this kind of interlocked, kind of dependent relationship between the contact network, the transmission network, and the phylogeny. So I guess first it's good to kind of step back and think, okay, well, the contact network, it's just some network. It's a graph where you have nodes representing people, you have edges representing risky contacts across which a pathogen could hypothetically transmit. And as you can imagine, the contact network is going to vary in structure quite significantly between different pathogens. So, and even within the same pathogen between different modes of transmission. So for example, if we're talking about like HIV sexual transmission, we might expect a power law degree distribution where there's few people with a lot of connections and there's many people with few connections. If we're talking about something like SARS-CoV-2, maybe it's a more densely connected network. But as you can imagine, the structure of this network kind of varies depending on what types of social interactions you're trying to capture across which the pathogen can transmit. But then, previous one real quick. And then given a contact network that you've randomly generated, the transmission network is also some random process kind of conditioned upon the contact network. So different pathogens and specifically different modes of transmission are going to have different rates of transmission. Maybe individuals can be in different states depending on whether or not they have treatment, whether or not they've been diagnosed, et cetera. So just how a pathogen transmits across a given contact network also has to be modeled in some complex way. Okay, so for our purposes, and this is actually something that Ravi developed, the way that we're simulating contact networks and transmission networks, so Natasha had alluded to the fact that prior works typically make a very unrealistic assumption that social networks are unchanging. So over maybe a 10-year simulation, whatever were the sexual contacts or whatever types of network that we had at the beginning stays fixed for the entire period, which is extremely unrealistic. Some partnerships form, some partnerships break, people might enter, people might leave. And then along that dynamic contact network, we need to be able to then also simulate the spread of the transmission events across that changing contact network. So Ravi developed this kind of two-stage contact network and transmission network simultaneous simulation software where we're modeling individuals that could either be in an MSM sexual network, heterosexual network, and injecting drug use network. And I want to emphasize that these networks are all overlain. So individuals can belong to multiple of these networks simultaneously. So individuals begin in stage 1, which is HIV negative, and then eventually they transition to stage 2, HIV positive. They could be either diagnosed or undiagnosed. They could be treated or untreated. They could be virally suppressed or not virally suppressed. And individuals can kind of transition between these stages throughout the simulation. So an overview of some of the characteristics that we're taking into account in this model, in terms of the population, it's an open population, so folks can enter the population, folks can leave the population, for example, through death. The demographics that we're trying to model, we incorporate age, we incorporate gender, race, ethnicity, risk group. And then in terms of the transmission network, the actual transmission model, again, I want to emphasize the contact network is dynamic, so partnerships can form and break over time. And as I mentioned, we have men have sex with men sexual contact network, heterosexual sexual contact network, injecting drug use contact network. And then in terms of health stage for biomarkers, we have CD4 and viral load that we're keeping track of. And in terms of preventions, we are also modeling PrEP. And the care stages that folks who are HIV positive can be in, or I guess even before HIV positive, the stages are they could be at risk, and then they could be HIV positive but undiagnosed. They can then transition to being diagnosed, but not in care. and then they can transition to being in care. They could potentially leave care over time, and then people can enter viral suppression or potentially leave viral suppression. And ultimately, individuals can die, in which case they leave the population. So what's especially challenging is, okay, let's imagine that you solve this problem of simulating the contact network as well as a transmission network that's compatible by it. It turns out that the viral phylogeny, so how does the virus actually evolve over time? It's heavily constrained by the transmission network. So the transmission network heavily constrains not only the possible topologies of the viral phylogeny, but also what are the coalescent times, the actual times at which we have these branching events in the evolutionary tree. So how do you actually simulate a phylogeny that's compatible with the transmission network that you simulated? So I developed a tool called CoaTran. This is coalescent simulation across a transmission network. And what it does is you give it a transmission network, and then you define whatever model parameters related to the viral phylogeny, and it tries to simulate a compatible phylogeny. Then what we can do is, well, we have the ground truth real HIV phylogeny from San Diego. We have the simulated phylogeny that we've generated, and we can now compare the simulated phylogeny against the true real data phylogeny to then measure different properties of the tree. So maybe is the topology of it looking realistic? Are the branch lengths looking realistic? Basically compare how realistic does our simulated phylogeny look compared to the real one. And what we're especially interested in is, can we somehow incorporate this comparison of our simulated phylogeny as well as the simulated contact and transmission networks to then calibrate the model parameters so that we can hopefully match the real values that we're calibrating against as well as possible. A non-trivial aspect of this is we have to first think, what are the metrics? So, before you can actually gauge how good you're doing, you have to have some scoring metric of what are we determining as good. So a non-trivial part of this project was discussing with some of our colleagues, what are some different metrics, what are specific properties of the trees, what are specific properties of the transmission network that we can actually calibrate to. For the transmission network, we already had a good idea about that, but for the tree, it was kind of unexplored territory, and we had to determine that for ourselves. So for initializing the tree, we had a little bit of back and forth throughout this, where in the original, so Fabites is the framework that I developed quite a few years ago now to do this end-to-end epidemic simulation. And with Fabites originally, so from the transmission network, you have kind of individual phylogenies for each transmission chain. So each individual person that was HIV positive at the very beginning of the simulation seeds their own transmission chain. And the CoaTran tool is able to simulate a phylogeny compatible with each individual transmission chain. But those individual phylogenies are not independent. There's some global ancestral HIV phylogeny that somehow connects them. So in the original Fabites, the simulated tree kind of looked like a real HIV tree. And we had some way of simulating this, we call it a seed tree, this kind of ancestral HIV phylogeny that the leaves of that phylogeny are the individuals at the beginning of the individual transmission chains, and the ancestor of that phylogeny was the most recent common ancestor of all of HIV. So it looked like a real HIV tree when we did this, but when we used the different metrics that we determined might be relevant for this study, the tree metrics that we measured on the simulated data were not consistent with the observed phylogeny. So what we needed to do was basically figure out how to make that seed tree, that initial phylogeny relating the transmission chains together, we wanted that to look realistic. So the solution was, because our contact network and transmission network model is modeling real individuals in San Diego who happen to also be in that real data set, we could effectively seed the phylogeny using the real phylogeny that we inferred from the actual data set. So what we do is we take the true phylogeny from the real data set, prune out individuals who were sampled after the start time of our simulations, and then each individual in the simulation maps to a specific person in the real data set. So then we can kind of use that real tree as our seed tree and kind of stick each of these individual transmission chain phylogenies onto where they would belong in the real tree. So this type of approach didn't exist. So one novel aspect of this project was we had to develop a way of doing this in a way that was hopefully realistic and made reasonable model assumptions. So yeah, so what we do is we take the real phylogeny, and then the tips there represent individuals that were, so the purple, blue, and green represent individuals who at the beginning of our simulations were HIV positive. And then let's imagine on the bottom, now we have each individual phylogeny that was simulated for each transmission chain. So the purple box is the phylogeny simulated for the transmission chain seeded by the purple individual. The red one is someone who maybe wasn't in the original data set, but then was simulated, at time 0. They just weren't sampled in the real data set before our simulations, which exists. We have people that are diagnosed but don't exist in the data set yet, or at least were positive but weren't in the sequence data set, and then et cetera. And then blue and green also are 2 individuals. So we simulate all of those. And then what we can do is basically sample where in the phylogeny would make the most sense for each of these individuals to fit. It's not a deterministic exact, it's not, we don't stick it in a specific part deterministically. We are simulating this as well so that we can get random variation with different simulation replicates. But the procedure that we're using to simulate where they should fit is now inspired by realistic assumptions about where they would fall in the true phylogeny. So what we were able to do is then take our observed tree, take the simulated tree, the seed tree that we pruned up until 2019. And the metrics that you see in the left column, these are some phylogeny metrics. So number of cherries. A cherry is any internal node that has 2 leaves as its children. So basically, you can like cherries, like you have 2 cherries on a stem. You can kind of think of like each internal node that has 2 leaves on it. And then we normalize by the number of leaves. And the short answer is basically you can see that the observed tree from the real data and the simulated seed tree match up for this metric. Treeness is a metric of what proportion of the branch lengths are internal branches, which again seems to match up really well. The Collis balance index is a metric of the tree balance. So trees can either be balanced or unbalanced, and we're matching this as well. And Sack balance index is yet another tree balance metric. So the short answer of this, you don't have to necessarily know the nitty-gritty of what these metrics imply, but basically across multiple metrics that capture both the structure of the tree as well as the branch lengths of the tree, it seems like our simulated tree is now matching up pretty well. So we solved this challenge. We developed a novel flexible simulation framework that can create simulation models encompassing transmission and molecular evolution processes, and we implemented them in hopefully a scalable enough way, which I think we'll talk about a little bit later.
Yes, I think we should. Thanks so much, Niema. That was great. So moving on to our second challenge, Ravi, take it over.
Awesome. Thank you, Natasha and Niema, for that. As sort of Niema left off there is comparing the simulated tree to the real one. Well, a question is how do we do that? Because the simulated tree, we know all the information. It's simulated. We know who's infected, diagnosis times, and then we make the HIV sequences. But for the observed data, the true data, we don't have all information. We only have partially observed one. So how exactly did we go about comparing the simulated data that we have to the observed? And that's sort of what challenge 2 is about. And so thinking about what is the observed data that we have? So what we have is, so I'm going to just take a step back and sort of describe this picture. What we have here is that the circles are representing individuals. And these individuals are people with HIV, and they are sequenced. And the lines between 2 individuals or 2 circles indicates that their sequences are similar below a threshold. And here we're using 1.5% as the threshold. And so this is the data that we have, but obviously there are more people who are living with HIV, and including those are people who are diagnosed but are not sequenced, and they are also part of this kind of viral genetic network, but we don't get to observe them. And then furthermore, we also don't get to have sequences and observe where people who are undiagnosed fit into this viral genetic network. And in San Diego, about less than half of individuals, people who are living with HIV, are sequenced. So we have a fair amount of missingness. And so there is a question of, okay, so what we get is from the simulation is this sort of complete, beautiful data, but what we have in the real world is this incomplete data. So how do we go about and match these two things up? Well, there are two ways that we can do this. One is to take our simulated data and do some kind of mechanistic approach to sort of remove individuals from the simulated data to sort of match characteristics of the observed data, meaning who is diagnosed and who is sequenced. So that's one option. And we'll sort of go through that first. But the other option is to take the observed data and go and build a statistical model to sort of impute upwards. Is given this observed data, what would be the full viral genetic network look like? That means including everyone who wasn't sequenced and who wasn't diagnosed. So going with and we sort of tackled both of these approaches, but the way that the simulation goes is following the mechanistic approach. And so here, what we really need to do is understand um, who gets diagnosed, how long does it take, and then who gets sequenced. And we want to implement that into our simulation model that Niema described. So what we start off from the simulation is, once again, it's the complete picture, what we left off with. That means everyone who is undiagnosed, diagnosed with or without sequence, we get to see that whole picture. And what we do is then first is we figure out, okay, at a given time period when we want to compare it, for example, 2019 or 2020 or whatever, who is undiagnosed at that point. And so that's step one. And then we remove those from the picture. Step two is then who would be sequenced among those who are diagnosed at that time. And so then we can identify those. And so by slowly removing individuals from our simulated data, hopefully what we can do is have our simulated data match characteristics of the observed data. And that was what Niema was showing in his second to last slide, of showing, hey, these are different characteristics that sort of seem similar when we have followed this process. And to figure out who's diagnosed, who's sequenced and stuff like that, we used actual data from San Diego to figure out those probabilities and the length of time between being infected to diagnosed and diagnosed to sequenced. The other approach that we can go is going backwards. So starting with the observed data and basically how do we impute upwards? And so taking the observed data, what would be the full data look like? And so what we have developed is a novel approach that conducts Bayesian inference, which is a more approach or paradigm aligned with thinking about missing data on these partially observed networks. And just briefly, what this approach does or how it works is that it's kind of using a network model developed by members of this team called the congruence class model for networks. And that is a pretty flexible model that allows us to kind of build in our own assumptions to be able to do this imputation. And one way that it runs faster and allows us to look at very large networks, which are these sort of HIV viral genetic networks, what it includes hundreds, tens of thousands of individuals, is that we're leveraging advances in what is called the graph enumeration problem. And so we kind of overcome a lot of the intractability that has hampered previous Bayesian imputation approaches. And we'll give an example, not using San Diego data, but we have applied this to, as Natasha said, we're moving a lot of this work also looking at using it for Miami-Dade County. So that's the example we'll look at here. So, okay, so we talked about that there is missing data. So how does that missing data look like in Miami-Dade? So here we're only looking at men who have sex with men, just for a simplification, though the method would be applicable across, but just sort of just thinking about this particular population. And what we have here is characteristics about men who have sex with men, and particularly two characteristics, their race and transmission risk. So by race, we're looking at Hispanic of any race, or Black but non-Hispanic, white, non-Hispanic, or other non-Hispanic. And by transmission risk, we're looking at MSM who do not have a designation for injection drug use, and MSM who do. And what we have here is just a table showing who is sequenced, who has a sequence in Miami-Dade. So that's the first column is the proportion of people. So there are 12,000, a little over 12,000 people who are diagnosed and living with HIV, who are undiagnosed, or who don't have a sequence, and about 5,400 who have a sequence. And what we can sort of see is that the percent then who are sequenced by these different characteristics differ. For example, if we're looking at white non-Hispanic, about 22% of individuals have a sequence. Where if we're looking at Black non-Hispanic, about 38% of MSM in Miami-Dade have a sequence. So there are discrepancies or differences in the propensity to be sequenced by your race, and we see a similar kind of differences or differences in propensities to be sequenced by transmission risk, where if you're MSM and PWID, that you have a higher propensity of being sequenced at 36 versus 30%. And so what does that missing data do? It not only gives us an incomplete picture, but it gives us a biased picture, right? Because if you have more people who are sequenced of a particular group, it may look like they have much more transmission or spread because you have more data on them. And so what we wanted to do is develop an approach, this Bayesian imputation approach on partially observed networks. And well, how do you test if it actually works well, a new approach? Well, one way to do that is using simulation studies. Because obviously, if you don't have the data, you can't really assess how well you do. So what you do is you create simulated data, and you do this subsampling, as we have sort of seen in observed data, and see how well your approach can reconstruct the simulated data. And that's what this diagram is kind of showing. And I'll walk through it kind of quickly. But what we did is what we were interested is understanding the number of connections in the viral genetic network that somebody has. And that's along the x-axis. So we have people who maybe are unconnected, that would be 0, all the way up to people who have about 15 connections. So you can see 0, 5, 10 as the markers. And the number of people who have those connections are on the y-axis. So in the simulated full data, this is our simulated data, so we know exactly what the network looks like. And what we do is we subsample and only look at 30%. And that's matching about roughly of the observed amount of data that we get in Miami-Dade. And so now you can sort of see that you get a lot of people who have a degree 0 all of a sudden. And that's because you're only seeing 30% of the data. But when you're thinking about what does 30% of the data really mean, to get a linkage, you have to get both ends. So you really only have 30% times 30%, which is 9% of your actual network data. And so a lot of people end up being unconnected. And so that's what the observed data looks like, is this high inflation of unconnected and a depressed kind of distribution across their people with different degrees. And what our imputation does is try to impute this and correct for this 30% or of observed data, So our 91% of missing network data. And here's the degree distribution that we're getting back. So the number of people who are unconnected in the viral genetic network, all the way up to 15 connections. Not perfect, but better than the observed. And that's always the hope and the goal. And actually, just eyeballing it, I think it doesn't work too bad given the level of missingness that we anticipate. Okay, so what does that mean in terms of now when we apply it to Miami data? And so here are two sets of results. So the first one, all the way on the left, there are 4 kind of stacked bars, and that is the observed data from Miami-Dade. And I'm going to go with the first stacked bar just to explain it. And so the first bar on the Miami-Dade is for Black individuals and their propensity to have a linkage to the other race/ethnicities. And so what we can see there in the first bar is that among Blacks, about 55% linked to other Black individuals in Miami-Dade. And about, I want to say 25 linked to Hispanics, and the rest are small to other and to white non-Hispanic. And then jumping over to our estimate, we can see that once we impute and adjust for these differences in sampling rates, we have a very different picture. In particular, we see that there is much more mixing among the races than what we would expect from the observed data. In particular, we see lower amount of Black linkages to other Blacks from 58 to 47. And we see lower to Hispanics from 37 to 15, and an increase in white and other races. And that's because, once again, what we saw in the Miami-Dade, why this sort of makes sense, or intuitively where you might get this, is that Blacks had a much higher propensity of having a sequence compared to whites, for example. So when you sort of adjust that, you would sort of expect that you see an increase of linkages among whites when you sort of adjust for these differential propensities of having a sequence. And yeah, overall, you can just sort of see that that is true across that. We see a lot more mixing among the races, which, and so that's sort of how we are tackling challenge number 2.
Thanks, Ravi. All right, so in our last few minutes, I'll talk about the choice of the calibration metrics, and then finally, what does this all mean? What do we get out of it? So as we've been mentioning throughout this talk, there were a lot of different metrics that we could compare, and there are really no established methods for what metrics we should use to compare our molecular data with our epidemic model. So at first, we started with looking at what Ravi has just shown you. The distributions of the proportion of the genetic linkages by transmission risk group. You can do this by race, ethnicity, transmission risk group, et cetera. And other studies that have looked at molecular data, like Joel Wertheim's and others, have looked at this metric. And so we thought we'd start with that. But there's a lot of other metrics that you could think of to use. So I won't be showing you results for these, but for example, we may be interested in using characteristics of growing clusters and how those vary. Because we see that there are different characteristics in the data about growing characteristics by race, ethnicity, and exposure category. We could also look at cluster growth by geography. So Antoine Chalion and the rest of the molecular epi team has done this analysis in San Diego County to look at cluster growth and geography, and really interesting analyses here. And he's also done some really fabulous work looking at movement and phylogenetic. So thinking about dispersal events between locations and between risk groups. Here, some really cool work looking at viral migration patterns between San Diego and Tijuana overall, and then among specific risk groups, which is really fascinating and maybe something that we could use. And similarly, kind of looking at relative contributions of migration events. So of all the events that appear to be events where the virus has moved from, for example, one location to another, or from one risk group to another, can we use that information in terms of calibrating the model? So there's a lot that we could do. And there's a big question about, what is useful, what is valuable, and what doesn't really help us answer our problem. So this is kind of exploration in progress. I think, honestly, we'll be doing this over the next few years with our Miami project, trying to understand this in more detail about how these metrics could be useful in our project. But okay, so then we've developed this really amazing, innovative methodological engine. And so what does it all mean? What do we get out of it? And so, the project is really assessing the potential value of incorporating this data on projecting HIV incidence. And so I'll show you some preliminary results today. That's really kind of a case study, a sort of test case. So trying to say, well, does this help? And what does it say in the end? So what we do is we simulated HIV transmission in San Diego County, and we do two separate simulations. We do one simulation where we calibrate the model to epidemiological data only. So for example, new diagnoses by race and ethnicity, and ensure that our simulation matches those data. And then we do a separate simulation model where we calibrate to both the epidemiological data and the genetic data. So these viral genetic linkages by group, which I showed you in the previous slide. And so we've got two calibrated simulations. And for each of the simulations, so one I'll call epi only, it's calibrated only to the epi data, and one I'll call epi and genetic if it's calibrated to both epi and genetic. And for both of those simulations, we assess, well, how well does the model fit to the observed data and predict the incidence in the future? And are there differences in the model predictions in terms of the impact of intervention scale up? And in this talk, I'll show you an example where we simulated a hypothetical intervention of HIV testing scale up among men who have sex with men, just as an example. All right, so here are some preliminary results. So we have these two simulations. So what this slide is showing you is the blue dots and lines are the observed data in terms of new HIV diagnosis over time. Each of these squares is how many new diagnoses we have each year by transmission risk group. So among people who inject drugs, men who have sex with men who have sex with men who report injection drug use, and other. And then we have our two calibrated simulations. Our green is our... I can't see because it's covered. One is our epi and genetic, and the other is our epi. And so we have our two calibrated simulations, and you can see that both of our simulations calibrate well to the observed data that we have. So both our red and our green simulations are matching our blue simulations pretty well for the number of new diagnoses over time from 2019 to 2021, where we have data. And both of them are predicting fairly similar trajectories over time, maybe a little bit of differences, but you can see these bars indicate the uncertainty in our estimates because we run multiple replicates over time, so there is uncertainty. And in general, both are calibrating well to our data and new diagnoses and predicting similar trajectories over time. But each simulation is very different in terms of how they calibrate to the genetic linkage data. I know this is a complicated slide, but effectively on the left-hand side, what we're doing is we're taking that stacked bar chart that you were looking at in terms of the genetic linkages between risk groups that Ravi was just showing you, and I'm just converting that stacked bar chart into a bar chart. And effectively what we're seeing here, the blue is our observed data. So the observed proportion of linkages between these groups and the observed data is blue. And the orange is what the calibrated epi-only simulation shows. And you can see that, there's a pretty large differences in terms of our observed data and our simulated data for our epi-only calibration. Whereas on the right-hand side, you can see that there's a much better match between the observed data and our simulated data for our model, where we both calibrate to the epi and molecular data. So, I mean, this isn't a huge surprise. Obviously, the model on the right-hand side is calibrated to the genetic data, so we would hope that it matches better. But it's a proof of principle that, we don't get a good fit if we're not calibrating to the genetic data, and we do get a good fit when we additionally calibrate to that data. And what we can see is that it indicates oops, sorry. Let me just go back. And so what does it do? Well, it gives us information about parameters, in particular, the proportion who have multiple risks, so men who have sex with men and women, and sexual mixing patterns by risk group. So because we're additionally calibrating to these data, we're getting additional information about our populations and the mixing between our populations, because those calibrated parameters are different between the simulation on the left and the simulation on the right. And so here's what the model is projecting about true new HIV infections. So before I was showing you HIV diagnoses, because that's what we can calibrate to observe data, this simulation is showing the simulated projections in terms of new HIV transmissions over time by risk group. And without any intervention scale-up, you can see that these two simulations, the epi-only and the epigenetic, have pretty similar predictions in terms of epidemic trajectories over time. This is without any intervention scale-up. But with an intervention scale-up, so if we increase the diagnosis rate among men who have sex with men only, we actually start seeing differences between these simulations. And it's kind of hard to see here, but I'll show you on the next slide. So what we see here is this is the relative reduction in HIV incidence from 2024 to 2030 with our intervention scale-up. Comparing our simulation with the epi calibration only in the left, in this sort of pink, and the epi and genetic in the sort of blue-green. And so both simulations predict a pretty similar impact of this intervention on new HIV infections among men who have sex with men only. Here you can see about a 15% reduction in HIV incidence estimated. But we start seeing differences in terms of the impact of this intervention among other groups. So for example, we're predicting a 27% lower impact among men who have sex with men who also inject drugs in our simulation, where we're more accurately capturing the genetic information and we believe, more accurately simulating the mixing patterns between groups. And that was because we saw a different levels of mixing than in our epi only, where we weren't informed by that. So we're starting to see predictions diverge and different levels of impact among these other groups. All right, so overall, I think we have successfully developed a really novel methodological framework to incorporate molecular data into model calibration of these simulation models to inform prediction of HIV incidence and the impact of interventions. The work is in progress. We're still refining model calibration. This is incredibly computationally intensive. We've had to move servers because we ran out of money on the UCSD server. We're now running it on a different server, and it took a month to kind of get that up and running. So part of what makes this work slow is that it's incredibly computationally intensive, and Niema's expertise in terms of speeding up how we do this work is going to be critical in terms of, ensuring that we can run these simulations efficiently and really explore all of the different areas that we want to explore. We're in the process of implementing PrEP scale-up, which is, I think, a realistic intervention that probably will scale up over time to kind of make more accurate, specific predictions in terms of HIV incidence in the future in San Diego and in Miami-Dade. And we want to look at other molecular network features that could be informative or potentially more informative for HIV incidence estimation. And obviously, ideally, we would do model validation. And so, we'd like to either do work, hopefully we'll have longitudinal data, especially from Miami-Dade, to sort of see whether we can validate our model against future incidence estimates or future estimates by subgroup. As I mentioned, this work is really flying now because we're applying it to Miami-Dade County, which is a NIH-funded R01 that Susan and I lead. And ultimately, we'll be applying this really in a resource allocation way. So I've shown you predictions today in terms of HIV incidence. What we're doing in Miami-Dade is we're actually collecting local programmatic HIV program data in terms of costs and impacts. We're incorporating geography, and we're going to ultimately use this to help Miami-Dade Health Department understand how to optimally allocate their HIV prevention resources, either by group or geography, in order to maximize benefits and improve health equity. So thanks again, huge team that's been working on this. I just want to emphasize this has been one of the most exciting projects that I've worked on in terms of really bringing together a lot of heavy lifting quantitative and analytics work to make this actually run. And then the Miami-Dade project, because it's incorporated a lot of important information on health equity and how we're considering these populations, has relied on experts in terms of qualitative research, and the HES core and the translation of virology core and the BAM core of the CFAR is really a nice collaboration through this work. So thanks to all that have been involved, and very happy to take questions. I see we have a few in the chat. There's one about how social networks might be factored in antecedent to the sort of sexual and injecting transmission networks. Ravi, I'm not sure if you want to comment about that.
Sorry, I'm going to just clarify. That was me. It's Nettie. Hi. That was so interesting. And I was trying so hard to follow, and I didn't have time to really chat the question very well, and we have limited time. So it might be for next time. But I was just thinking about, before you have a contact, you have a pre-contact, and you by having that pre-contact change your chances of being an incident case, even if you don't have sexual or direct transmission with that person, right? So when you meet your drug-using boyfriend, then you increase your chances of joining the user of drug cohort, even if you don't use drugs with him. So how do you think about that part? And it might be for a different conversation, but thank you.
I think it's a really fascinating question, and I know Britt has done a lot of, fabulous work looking at social contact networks, and in fact, how self-reported sort of social contact network can give us additional information in terms of, the injecting and sexual networks. So it would be a really cool area. Not something that we've looked at yet, but I think could be very interesting. Ravi, do you want to add?
No, I think I just agree with you that it's a very interesting way to sort of think about how do you meld this additional piece of information, and how does your social context inform your behavior at present and in the future. So I think, yeah, great question, not a great answer on my part.
And I'm sorry we didn't leave enough time for a lot of questions. I know people are probably needing to jump off. So thank you so much again for coming, and very happy to stay on to answer questions if people have them and have the time.
I would also say you could email the crew as it is at the top of the hour, but I love that you just wrapped up for me and us. That was great. This was really fabulous. Thank you so much.
Thanks so much. And yeah, thank you,
yeah.
Nettie. Sorry, can I just jump in? Because Nettie put in the chat. We would love to come back. As I mentioned, Chadwick has done some amazing qualitative work in Miami-Dade in terms of this idea and the potential risks and benefits and stakeholder perspectives. And right now, our team is kind of working with him to think about how to incorporate those perspectives into our work. And it would be great. We'd love to come back and kind of give an update on that iterative piece of work as we work through the sort of risk benefits, ethical questions, as you say, of when we can use the models and how to use them properly, et cetera. So very happy to come back and talk about that as we work.
Thanks all.
Thanks all. Have a great weekend.
Have a great weekend.
Thanks for having us. Thanks, Ravi. Thanks, Niema.
Bye.
Thanks, folks.
Bye.